What I want to do today is to take you through a fairly whistle-stop tour of some of the things that you can use this technique for when looking at laboratory studies, and then to make some suggestions for what we already know can tell us about what we might need to do in the future. So why do we do research? Well, in terms of laboratory research, we do it first of all to understand what causes the disease we're interested in, and in vitro or in vivo research to try and understand what biological processes might be important and which might be epiphenomenon or less important. And then we would like to be able to change these processes in experiments and then change outcome in animal models of our diseases. And also from animals, we might like to know that our treatment is probably safe, with the limitations of safety in animals not being the same as safety in human animals. And then we'd want to show in clinical trials that the treatment changes outcome in our clinical trials, and then know that the treatment actually works in the real world. But I want to show you that science can be wrong. This is probably my favorite paper of all time. I saw this first as an abstract at Neuroscience in Miami in 1997, where they showed that homeopathic doses of glutamate and arnica montana improved outcome in a focal ischemia model, and these data show that half-dose, low-dose glutamate improves outcome a bit, and full-dose, low-dose glutamate improves it more. And you could show graphs like this for any animal study of any drug over the last 30 or 40 years. But when I tell you that half-dose, low-dose glutamate is 10 to the minus 120 molar, and full dose is 10 to the minus 160 molar, where first of all, the homeopaths have their columns the wrong way around, because this should be more powerful. But secondly, those of you familiar with Avogadro's constant will know that there isn't anything in either of these columns. So there's something wrong, I would argue, with the model, rather than homeopathy actually being alive. This is probably my second favorite publication. This is the Atlantic salmon, which many of you will know. If you do functional MRI imaging, it's important that you know that your scanner's working. And a group in the States used to have a competition to see who could bring in the most outrageous organic object to scale their scanner at the beginning of each week. And there was a fish market on the way that one chap came into work. And so one day he picked up an Atlantic salmon and he put it in the scanner. And then they thought they would try and find out what happened when they tried to get the salmon to do something. Because this is what happens in all of these studies in high-impact journals of functional MRI imaging. So they took one mature Atlantic salmon, it was approximately 18 inches long, weighed 3.5 pounds, and was not alive at the time of scanning. The task administered to the salmon involved completing an open-ended mentalizing task. The salmon was shown a series of photographs depicting human individuals in social situations with a specific emotional valence, and were asked to determine what their emotion was. And then they scanned the salmon before and after each exposure and put it through their standard image processing. And lo and behold, they found that a bit here in the salmon's orbitofrontal cortex lit up. And this is because they were measuring something like 8,000 voxels, and it's not surprising that some of them light up. And this was, at the time, the processing that was standard for functional MRI studies with normally 5 or 6 human subjects per group. This is what I used to struggle with when I was a bench scientist, trying to understand a pathophysiological pathway starting with something like p53-dependent apoptosis and ending up with either neuronal death or neuronal survival. And any TINS review has got 3 or 4 of these pathways. When you try and understand the results from a gene array experiment, for instance, you get kicked out perhaps 20 genes that are regulated up or down by your condition of interest. And there's a tool called ingenuity pathway analysis, which then tells you if these are linked and how they're linked. And these colleagues from Edinburgh were interested in changes in a neuronal cell line undergoing apoptosis, and they were delighted to see that their results showed something that was statistically significant at the level of 10 to the minus 16 or 17. But they looked at the genes, and they thought that they couldn't see what the links were. So very cleverly, I think, they took another 13 proteins selected at random from the genetic basis and put it through the machine. And then they took another, and another, and another, and they bootstrapped 1,000 randomly selected sets of genes. And they took the ingenuity pathway analysis score, which is a logarithmic log odds score. So these ones here, this is supposed to happen by chance. This one here, 1 in 1 million times. And none of these 1,000 sets were called as not being significantly associated. And the median significance level was something like 10 to the minus 18. So what this tells you is that genes are connected one with another. It doesn't tell you anything about your situation in your experiment, and it's completely useless to take you further. So what I want to do is talk a bit about systematic reviews of the efficacy of candidate interventions in animal models of disease, then give you some results from overviews of a disease model taken more generally, because I think that can be important, then talk a bit about some secondary analysis, and then about some future directions. We've already had a reasonable definition of what a systematic review is, but importantly here, I think this is taken from that fount of all knowledge, Wikipedia, and it may have changed. But systematic reviews are quite common where an assessment of methodological quality for precisely defined subject would be helpful. And that's one of the principal purposes that we've used that for. So for our FK506 systematic review, Michael, knowing with precision, on average, how good FK506 is in an animal model of stroke isn't that important, but knowing that this set of studies are much less effective than these set of studies with a different characteristic, that's the kind of gold dust that we've been using. I should give you a caveat, though. And that's that this is observational research, not experimental. These are the 50 states of the United States of America, and they're ranked according to the average IQ as determined by the American Psychological Association, and they're color-coded as to their voting behavior in the Electoral College in the 2004 presidential election, with these Democrat states being blue and then clever, and these Republican states being red and less clever. Now, you and I believe that there's a causal link between average intelligence and voting behavior, but this doesn't tell you this. This simply tells you that there's an association, and that's an important caveat through everything that I'm going to show you. So let's look at what we've learned from reviews of specific interventions, particularly the low prevalence of measures which might be used to avoid bias. And we've been through these in some detail, but selection bias, where animals differ before the experiment starts, that you would get round by randomizing them. Performance bias, where the investigator does something differently for the animals that are getting treatment, for the animals that aren't. Pays them more attention, is nicer to them, makes sure that they take the filament, including their artery, out before they go for coffee, not afterwards. But if they didn't know which animal was getting treatment and which wasn't, you wouldn't have that problem. Detection bias, where they call the outcome differently, you would get round by blinded outcome assessment. And attrition bias, which you might get round by reporting dropouts or intention to treat analysis, which is done so rarely in animal studies that we've not been able to look at it further. But this is data from brought together in a review that we did for TINS a couple of years ago, looking at, on the same 10-item checklist that you saw earlier on, the prevalence of publication, peer-review journal, statement of control of temperature, important in stroke modelling because of the protective effect of hypothermia, down through randomisation, allocation, concealment, blinded assessment of outcome. And for the E's on your eye, green is good and red is bad. And overall here, we found, for instance, randomisation in only 36% of studies, allocation concealment in 11%, sample size calculation in 3% of studies overall. These are our cause for substantial concern given what we think we know about study quality. Let me show you how it really is a cause for concern. Because if we then dichotomize studies, these are hypothermia studies from Bart, who's at the back, review of hypothermia, into those studies where the allocation to group was done at random or not, or where the assessment of outcome was done blinded or not, you can see there is a substantial and significantly lower efficacy in these high-quality studies. Still substantial efficacy remains, and this is a huge data set, but significantly lower. This is a review of a drug called NXY059, which was a candidate neuroprotectant test marketed by AstraZeneca under the name Cerevive because it was held to make your cerebrum survive until their second phase 3 clinical trial showed that it was inert. The clinical trial program was based on only 408 animals, which I would argue is far too few. There was a substantial improvement in efficacy, but when we looked at randomized studies, allocation concealed studies, and studies with a blinded assessment of outcome, the efficacy was falling away. And not one of those 9 studies, only 9 measured infarct volume, not one of those 9 studies met all of these criteria. And the 2 studies used by AstraZeneca in their investigator brochure met none of these criteria. So there's a problem with those animal data. And then there's the issue of how you design your clinical trial. So this is NXY059 again, and this is the efficacy in normotensive animals, and this is the efficacy in hypertensive animals. So in rats with hypertension, NXY is much less effective. 7% of animal studies had animals with hypertension, but 3/4 of the patients in the phase 3 clinical trial that was neutral had patients with hypertension. So they hadn't tested this in the animal population that was most like the humans in whom they wished to do the clinical trial. These are the TPA data, and Michael showed a different slide from this paper earlier on. And again, you can see that TPA is much less effective in animals with hypertension. But the clinical trial specifically limited entry to patients without high blood pressure because they were concerned that it might make their artery pop when you unblock the blood vessel. So the clinical trial was done on the patients who were most like this group of animals. And if we look at time to treatment in stroke, pivotal. TPA works in stroke, and Torilizad appears to work in animals but doesn't work in human stroke. In the animal literature, the median time to treatment for TPA is about 90 minutes, and in clinical trial it's about 90 minutes. For Torilizad, the median time to treatment was about 10 minutes, and it was more than 3 hours for more than 3/4 of the patients in the trials. And so if you look at the TPA data and look at the decline in efficacy over time in this animal cohort here and in this human cohort here, you can just about put those decay curves on top of one another. And Ian and I were talking earlier on about corticosteroids for traumatic brain injury, where in the review that he did, the corticosteroids were given, we think, about on average 5 minutes after the induction of the brain injury, which, nice to know that it works, but clearly you're not going to be able to get any treatment into any human within 5 minutes of their head injury having occurred, unless you were the one who did the head injury. So reviews of individual interventions, I would argue, show first of all that there's a low prevalence of those measures which might help secure the validity of those experiments, that high-quality studies give lower estimates of efficacy, and that there are substantial avoidable differences. Of course, an animal model doesn't tell you what will happen in a human, but there are some things you can do to an animal that might make it more reflective of what might happen in a human, and those tools are rarely used. Let's look now at systematic overviews of disease models, and let's start off with EAE. This is Hannah, who's in the audience, who's got some graffiti, some horrible graffiti on her cheeks, but she looked at animal models of experimental allergic encephalomyelitis, the animal model of multiple sclerosis. And what she's done, you don't want to come and do a PhD with me, she looked and started off with almost 10,000 publications, which she started to screen down. And the reason for showing this is that there are a number of publications here which were excluded from down to 203, reporting either a mean maximal severity or a mean clinical severity for behavioral outcomes. 72 didn't have a valid outcome that we could measure. So 58 didn't give any estimate of their variance. 7 didn't tell you how many animals were in each group. Some had uninterpretable, and by uninterpretable data, I mean take it to an EAE doctor and say, what does this mean? And they go, I don't know, I've read this 5 times, I don't know what they're saying. But she ended up with 126 publications reporting a mean maximal severity, a mean clinical score for one of 36 drugs tested 5 or more occasions with a valid outcome measure that we could take. And what she found, again, was that randomized studies gave lower estimates of efficacy, blinded studies gave lower estimates of efficacy. If you looked at the sample size, and this is a discussion we were having yesterday, I think sample size is a direct cause of bias in these very, very small animal studies. You can see that in the smallest animal studies, the reported efficacy is high, and in the larger studies, that efficacy gets much, much lower. And I think that's because there's a reporting bias that these are more likely to get reported than these. There's another issue about the generalizability of the EAE literature, which is that when you give an animal EAE, you inoculate it with something which stirs up their immune system, which then goes on to attack their myelin. And they develop the behavioral phenotype at about 10, 11, 12 days after that has occurred. At that stage, that's when the patient's general practitioner is making an appointment for them to see the GP, and then they see me about 2 weeks after that. So the earliest I can treat someone with this condition is probably about 4 weeks after that inoculation. But you can see that in the EAE literature, 2/3 of studies start treatment either before the EAE is induced or on the day of EAE, and the efficacy there is quite good, but beyond 2 weeks, which is just when these patients would be coming to clinical attention, or developing their symptoms even, the efficacy is substantially lower. So I think in EAE, we're developing a whole lot of treatments which influence our ability to develop EAE in an animal, rather than something which might be of use in human disease. And Alzheimer's disease, Kieran is here as well. He's looked at studies, all studies that we could identify in the literature that had used a transgenic model of Alzheimer's disease to test the effectiveness of a candidate drug. And again, he started off with about 8,000 publications at the top and boiled this down to 250 odd, looking at plaque burden, outcome, which is a histological outcome, or outcome and performance in the Morris-Water maze, which is a test of learning and memory. And again, had to exclude a number of studies because methodologically they were uninterpretable. But what he sees in the Morris-Water maze is in this absolutely hugely well-established model of learning and memory used throughout the neurosciences, is that most studies did not blind their assessment of outcome as the rat tries to find the missing platform. The mouse actually tries to find the missing platform, and those where the investigator didn't know whether the animal had received a drug or not gave a much, much smaller and barely significant improvement in outcome. A real cause for concern in a technique that's used throughout neuroscience, not just in drug development. Parkinson's disease, almost 12,000 publications, excluding 83 because no control group, no variance, not telling you the number of animals per group, but looking at the efficacy of dopamine agonists in Parkinson's disease. This is blinded assessment of outcome against a number of different behavioral outcomes, and you can see that the high-quality studies consistently give substantially lower estimates of efficacy, and on a composite quality score, in this model of lesioning, which is taught, or I was taught as a pharmacology undergraduate, was a bioassay of the efficacy of a dopamine agonist, that you lesioned one side of the striatum, gave the animal a dopamine agonist that made the animal run round and round and round, that in high-quality studies, this effect disappears completely. So it may be that it doesn't exist, that people have just convinced themselves that this happens, and so they see it because they think it's there. So a low prevalence of measures which might help secure the validity of the work done. Many studies don't describe their data adequately, and the intrinsic limitations of animal models, to my mind, are amplified in the way that they're used. Rather than being addressed in the experimental setup, they're just used in a way that helps get the result that's significant, because I've managed to reverse EAE if I give a treatment before I give the drug. Well, so what? That's not really the best way to try and develop drugs. This is the prevalence of some measures to improve validity across a number of research domains. We've done stroke, group in the States have done motor neuron disease, Alzheimer's is Kieran's work, PD and EAE you've seen, glioma we've had students doing in the past, and this is Gillian who's embarking on a Herculean task looking at pain. And you can see that, if anything, those prevalences for stroke that gave us cause for concern represent actually the best that animal research does rather than the worst. But we're at 36% for randomization for stroke, going down to 8% for EAE, blinded assessment of outcome 29% for stroke, but 0% for glioma, and an astonishingly low prevalence of sample size calculations. And this is work that Hannah's done, looking at a year in the life of the Journal of Cerebral Blood Flow and Metabolism, with the consent of the editor, looking at the prevalence of reporting of randomization, allocation, concealment, and blinded assessment of outcome, but dividing it into in vitro work, animal work, and human work. The point of this is that animal work is actually, if anything, slightly better than the in vitro work. So I don't think we should restrict our focus to animal models of disease, because I think the entirety of the life sciences is probably confounded by a low prevalence of taking simple precautions which might improve the validity of your results. And this is the NC3R's Carol Kilkenny paper from 2009, where they looked at 271 papers funded by NIH or MRC, and found sample size calculations in none, randomization about 12%, blinding in only 14%, and you've seen the JCBFM data already. And that's led to the development of the ARRIVE guidelines, and there are other guidelines, which are all broadly similar, I think, in the way in which they set standards for the reporting of animal studies or of laboratory research. But I would counsel that reporting isn't the only thing that's important, and we need guidelines on how people should actually do the research, not just on how they should report it. But I think this and others are important contributions. For secondary analyses of large data sets, well, we've heard already about publication bias, and we almost missed that picture. So I've shown photographs of all the team. I emailed them a week ago saying, please send me a photograph for my talk, and if you don't, I'll use one that I've got already. And this is a very young Dr. Emily Sena, I'm told, and David will appreciate, this is her father, I think, and he's got a glass of Shiraz in the other hand, which is what she's reaching out for and something. Anyway, Emily did a fantastic job looking at publication bias in stroke models and found, first of all, only 2% of publications had no significant treatment effect, slightly lower than one would expect to occur by chance. About 16% of studies remained unpublished, and this resulted in an inflation in efficacy from an industry standard 31% improvement in outcome was really substantially less than that if you accounted for all the neutral studies that were missing, that were likely to be missing. And here you see them for the individual drugs, and even for hypothermia, which, as Bart knows, is the best thing since sliced bread, there is still a substantial publication bias. This is Kieran's data for the preclinical Alzheimer's disease literature. You've seen this before, and as Michael pointed out, there are a large number of negative studies here which are missing, and this is statistically significant and substantially reduces the estimates of efficacy. There are other lovely things that you can do with systematic review. There's a dogma in the Alzheimer's literature that some people believe that the two fragments of the amyloid beta protein which cause the disease are equally important. But there are a group of fundamentalists who believe that amyloid beta 42 is really important and amyloid beta 40 is irrelevant, and that these are regulated separately. So what Kieran's done here is he's taken studies that have given a drug to a transgenic animal where they've reported both a change in amyloid beta 42 and a change in amyloid beta 40. And he's plotted each of these dots as an individual experiment, and then he's regressed them, and his high correlation coefficient here shows that actually these are measuring exactly the same thing. And the fundamentalists who say, I've got a particular piece of knowledge here that nobody else understands why this is important, well, the answer is that it's not. And if you were to look here at something like 70 or 80 individual studies, you would see that there aren't any differences of note. The development of EAE, it was always held that the Th1 cell was important in this development, but then along came the Th17 cell, and a group of fundamentalists said this is important and that's not. So what we've done here is we've looked at studies that have reported a change in the severity of EAE and a change in the number of the population of Th1 cells, and these studies here are concordant. They agree, they say the same thing, and these here are discordant. And what we've been trying to develop is a way of summarizing that information where dots on the line show concordance and off the line show discordance, and the size of the dot is the number of studies and the color of the dot is the quality, so you can see that the quality is poor, but there's probably more evidence to support this pathway than there is to support this pathway. So you can use it to understand pathways. You can use it to try and understand a bit about surrogate outcome measures. This is self-evident, but in stroke we'd like to be able to measure something half an hour after I've had my stroke that'll tell me whether my treatment's going to work, instead of having to wait 3 months to see whether I need a wheelchair, because that will help me refine my intervention. So is there a surrogate outcome that I can test for my drug? So what we did here was we took drugs where we'd got a report of both a functional and a structural outcome in the same cohort of animals, and we looked at how much of the difference in functional outcome could be explained by the observed difference in structural outcome. And what we should have known before we started was that it's different for different drugs. So the take-home message here is that there are some drugs which affect the development of the infarct, but don't have any effect on the evolution of the behavioral deficit. There's some interventions, like stem cells, which don't have any impact at all on how big the infarct is, but do affect what that results into the animal. And there's other interventions, hypothermia again, which seem to have effects at both of these dimensions. So the take-home message is that you can't use a surrogate outcome measure to refine your intervention until you know whether your intervention works. And if you know that your intervention works, you don't need a surrogate outcome measure. So it's all very circular. We've been looking, this is the question that you asked, this is about impact factor. Does high impact factor reflect high quality research? And those of us in the UK have a forthcoming what's called Research Excellence Framework, where we will be judged according to the excellence of our work, as judged usually by the journal in which it's published and the implied quality of that journal. So for 500 publications in stroke, we found a very weak association between the impact factor of the journal and the quality, using our quality metric, of the work published therein, whereby only 6% of the variation in impact factor was explained by the quality of the work. Looking again in stroke, the number of citations received by that publication driven by the number of checklist items scored, There is no association whatsoever between the number of citations and the quality of studies. So when your director of research comes along and tells you that you only publish in lousy journals and your work must be lousy, you can say, actually, there is no association between one and the other. And while it's very easy for you to come along and measure research output in this way, it's not a good measure, it's a lousy measure of research quality, and they should start again. It might be harder, it might be more difficult, but if it's really worth measuring quality, that's the way they should do it. This is a bit of fun. This is the National Self-Citation Index that we've developed. So this is a measure of the extent to which scientists in any given country are more likely to cite the work for their compatriots from that country than those from other countries. And if you cited at the average level around the world, you would have a national self-citation index of 1. And of course, everyone's going to do a bit. But what's striking about this was that those people that we always thought didn't let anyone else into their journals are actually very, very good at spreading credit and citing work from overseas. And we're not so bad in the UK, and I couldn't resist this. India and Russia are particularly bad at citing internally and not citing externally. And I think that's important. So what I hope, though, that I've been able to convince you of is that if you collect large data sets when you're doing systematic reviews and collect them in a way that you can pool those data, there's the potential for a number of really important, large, cross-disciplinary analyses that have the potential to give us new insights into the way that science works. And I think that provides a powerful argument for doing, as we've tried to do in Camarades, provide a single platform that these data can be collected in. So our data set now, I think, has data from experiments involving over 80,000 animals, something like 7,000 experiments, something like 3,000 or 4,000 papers, and it continues to grow, and that's what's driving and fueling these secondary analyses. So in future directions, I think we need to think about methodologies and refining these methodologies, because I don't think they're very good. I agree that there are big problems with the approaches that we use that are different from the concerns there are with systematic reviews of clinical studies, where large numbers of subjects in each group mean that your estimate of error is a good estimate of the error. If you've only got 5 or 6 animals per group, your estimate of the error is poor. And if you're using your estimate of error to drive your measure, which you do in some of these meta-analyses, that's a problem. We want to have more reviews of other interventions before we take them to clinical trial. We want to think about summarizing evidence from pathological pathways. So this is summarizing the data for stroke transgenics and the nitric oxide synthase for eNOS, iNOS, and nNOS, giving very precise, these are 95% confidence limits, estimates of the impact of that transgenic manipulation. I will skip through Hannah's work here, but essentially, in the development of EAE, inflammation results in a neurobehavioral score, and the question is how this gets to that. And this is using the same technique of looking at the effect size in one outcome, the effect size in the other, and seeing what the association is between them. And importantly, if you look at very early treatment, inflammation it seems to play a crucial role, and that's where a lot of the treatments are targeted. But if you look at late treatment beyond 7 days, inflammation seems to have very little role, and you should be more concerned about axon loss and neurobehavioral score. So this helps drive the focus for drug discovery. You can coming to a close now. You can use this in vitro research. This is lovely. This was done by a group that I didn't even know existed, and they're 200 miles away in Aberdeen. They looked at experiments reporting affinity binding constants at different cannabinoid receptors. And just to concentrate on this bit, these studies did not put in a protease inhibitor. So they chewed up a bit of brain and just left it on the bench and then measured the receptor. These put in an enzyme, these put in a protease inhibitor to stop proteins being chewed up. And these say that the drug has got very high affinity, that the receptors get very high affinity, and these say the affinity is in the micromolar range. And of course, what's happened in these studies is that the receptor has been chewed up by the enzymes, and so. But half of the studies in the scientific literature reporting this affinity don't take that simple measure to try and give them a more precise estimate. Pain modelling we've talked about a bit with Gillian, but I think in terms of experimental design, you can imagine that if you've got a pain model, you might have one that has got fairly low harm or distress to the animals, and one that's got a much higher level of harm or distress to the animals. And if you knew that you needed fewer animals for this one with low harm and it told you the same thing, then everyone would use this model. But the only way you can find that out is by putting together lots and lots of the studies and saying, well, what are the characteristics of this model across 100, 200 experiments? And I think this is one of our problems, that venerability and age counts for much more in science than it does nowadays in other walks of life. And there are many, many people who got where they got because they did what they did, and everyone assumes that if they want to get where their boss is, they should do the same thing. The first time as tragedy, the second as farce. There's a lot of talk about translational medicine, and if you look around the web, Googling for images of translational medicine, you'll get lots of fancy images about what it's supposed to involve. And what it seems to be, it's a bit like an airport terminal, that if things get complicated, rather than thinking about traveling a different way, you just add another terminal, and then you just add another terminal. Eventually it gets so complicated that people don't know what you're talking about. But I would argue that we need to be more rigorous in what we do, and we need to be more scientific in how we try and understand and explain where these translational failures have occurred. And we can do this by being rigorous in demanding real sample size calculations, by demanding the highest quality standards in the conduct and reporting of stuff. I've started now rejecting everything sent to me for review that's not randomized or blinded or with allocation. I just chuck it out. That's not good. Go. And everyone should do that, and then we'd all be able to get our good quality stuff into the journals. Model-specific GLP guidelines. I think there's important issues around a registry of animal studies, and where effect sizes are small, I think there's a lot of interesting work to be done to see whether we can develop tools and facilities for multi-center animal studies to pick up the sort of effects that, if true, would be very important in clinical trials. I think as scientists, science is the last bastion of people who aren't regulated, who don't have continuous professional development, who don't have to show that they're good at what they do, who don't reflect on whether what they do is appropriate or not. And so we should apply scientific principles and scientific methodologies to our understanding of where science is going wrong and what the weaknesses are of current research. That is to say, we should be scientific in the way we go about our science. So for all its exactness, it's one of the last professions where opinion and venerability count for more than evidence and rigor. And it's only when you analyse large data sets, as we have started to do, that you can start to say with confidence where people are going wrong. These large data sets can have multiple uses, and I think that they will be what drive translational breakthrough in some of these very difficult to treat conditions. And these are the people, I told some people yesterday about my slide showing people that hadn't funded me. These are the people who have funded me, and these are the people who did all the work. Thank you very much.